Imagine if every cancer biopsy could tell not only what the tumor looks like, but where it came from, which mutations are driving it, which drugs are most likely to work, and even how the patient is likely to respond to treatment. That is exactly what modern cancer pathology has become. Today, diagnosing cancer is no longer about looking through a microscope alone. It is about integrating morphology, immunohistochemistry, molecular biology, genomics, and artificial intelligence to deliver truly personalized medicine. The journey still begins with traditional microscopy, the foundation of pathology for more than a century. After a biopsy is collected, the tissue is fixed, processed, embedded in paraffin, sliced into extremely thin sections, placed on glass slides, stained, and examined under a microscope. Here, pathologists evaluate the architecture of the tissue, the size and shape of cells, abnormalities in the nucleus, how rapidly the cells are dividing, and whether the normal organization of the tissue has been disrupted. These microscopic features provide the first clues about the nature of the tumor and allow cancers to be classified into categories such as low-grade tumors, high-grade tumors, or invasive carcinomas. The first and most important stain used in pathology is called hematoxylin Eosin, or simply the H and E stain. Nearly every tissue biopsy examined anywhere in the world is first evaluated using this stain, making it the gold standard and the foundation of histopathologic diagnosis. Although the name sounds complicated, H and E is simply a combination of two complementary dyes. Hematoxylin stains the cell nuclei a deep blue or purple. Since the nucleus contains DNA, this allows pathologists to carefully examine its size, shape, chromatin pattern, and the presence of prominent nucleoli, features that often become abnormal in cancer. Eosin stains the remainder of the cell and the surrounding tissues in different shades of pink, highlighting the cytoplasm, connective tissue, collagen, muscle, and many other normal structures. Together, these two colors create an extraordinary amount of information from a single tissue section. Pathologists can immediately appreciate whether cells remain organized or have become disordered, whether they resemble the normal tissue from which they originated, whether they are invading surrounding structures, and whether they display cancerous characteristics such as enlarged nuclei, abnormal mitotic figures, variation in cell size and shape, and architectural distortion. In many cases, an experienced pathologist can make a diagnosis, or at least narrow the possibilities to a very short list, using the H and E slide alone. Yet appearance alone is sometimes not enough. Two tumors may look remarkably similar under the microscope, while behaving very differently or responding to completely different treatments. This is where immunohistochemistry, commonly known as IHC, becomes one of the most powerful tools in modern pathology. Immunohistochemistry uses specially designed antibodies that bind to proteins produced by cancer cells. You can think of these proteins as molecular fingerprints that reveal the identity and behavior of the tumor. When an antibody binds to its target protein, a colored chemical reaction makes that protein visible under the microscope. Suddenly, the pathologist is no longer looking only at the shape of the cells. They are also seeing what those cells are expressing at the molecular level. Some IHC markers help identify where a tumor originated. Cytokratins are found in epithelial cells, which line the body's organs and surfaces. Since most human cancers arise from epithelial tissues, positive cytokratin staining supports the diagnosis of a carcinoma. CD20 is a protein expressed on mature B lymphocytes and is an important marker for diagnosing B-cell lymphomas. S100 and HMB45 are commonly expressed by melanocytes and strongly support the diagnosis of melanoma. TTF1, despite its name, is normally expressed in both lung and thyroid cells, making it an invaluable marker for identifying primary lung adenocarcinomas and thyroid cancers. Other IHC markers do far more than establish the diagnosis. They directly influence treatment decisions. HER2, or human epidermal growth factor receptor 2, is a protein found on the surface of certain breast cancer cells. Some tumors produce excessive amounts of HER2, causing them to grow more rapidly. Fortunately, these cancers often respond remarkably well to targeted therapies such as trastuzumab, a drug specifically designed to block HER2 signaling. Without HER2 testing, these patients might never receive one of the most effective treatments available. PSA, or prostate-specific antigen, is a protein normally produced by prostate cells. When a metastatic tumor stains strongly for PSA, it provides compelling evidence that the cancer originated in the prostate, even if it has spread to bones, lymph nodes, or other organs. This information completely changes both diagnosis and treatment. Ki-67 is another extremely important marker. Unlike HER2 or PSA, Ki-67 is not used to identify where a cancer originated. Instead, it measures how many cells are actively dividing. A tumor with a low Ki-67 index is generally growing slowly, whereas a tumor with a high Ki-67 index contains many rapidly dividing cells and is often more aggressive. This helps physicians estimate prognosis and decide whether patients may benefit from more intensive therapy. Modern pathology extends even further by analyzing the tumor's DNA and RNA through genomic sequencing and molecular profiling. Instead of examining proteins, these technologies search directly for genetic mutations, gene fusions, and molecular alterations responsible for driving cancer growth. Some mutations have become essential therapeutic targets. EGFR mutations are commonly found in certain lung adenocarcinomas and can be treated with EGFR inhibitors. KRAS mutations occur frequently in colorectal and pancreatic cancers and are increasingly becoming targetable. BRCA1 and BRCA2 mutations are associated with hereditary breast and ovarian cancers and predict response to PARP inhibitors. TP53, often called the guardian of the genome, is one of the most commonly mutated genes across human cancers. Other clinically important alterations include ALK gene fusions in lung cancer and BRAF mutations in melanoma and thyroid cancer, both of which can be treated with highly effective targeted therapies. Genomic profiling also helps detect resistance mutations when cancers stop responding to therapy. In some patients, these mutations can even be identified through liquid biopsy, a blood test that detects fragments of tumor DNA circulating in the bloodstream, allowing physicians to monitor disease progression without repeated invasive biopsies. Perhaps the greatest advance in modern pathology is that none of these techniques are used in isolation. Instead, they are combined into what is known as integrated multi-omics analysis. Histology reveals the microscopic structure. Immunohistochemistry identifies protein expression. Genomic sequencing uncovers DNA mutations. Clinical information provides the patient's history and imaging findings. Together, these multiple layers of information create an exceptionally detailed biological portrait of each patient's cancer. This integrated approach answers numerous critical questions simultaneously. What type of cancer is this? Which molecular subtype does it belong to? Which mutations are driving its growth? Which targeted drugs are likely to work? How aggressive is the tumor? What is the patient's prognosis? No single test can answer all these questions alone, but together they provide an extraordinarily accurate diagnosis. Artificial intelligence is now adding yet another powerful dimension. AI algorithms analyze whole-slide digital pathology images containing millions, even billions of pixels. They can identify subtle patterns invisible to the human eye, assist with tumor detection, classify tumor subtypes, estimate tumor grade, predict prognosis, and even anticipate response to therapy. Importantly, AI is not replacing pathologists. Instead, it serves as an intelligent assistant, reducing observer variability, increasing diagnostic accuracy, accelerating workflow, and helping physicians manage the enormous amount of data generated in modern pathology laboratories. The complete diagnostic pathway has therefore evolved into an integrated workflow. A tissue sample obtained through biopsy or surgery is processed, stained with H and E, supplemented with immunohistochemistry when necessary, analyzed using molecular and genomic techniques, digitized for AI-assisted evaluation, and finally integrated into a comprehensive pathology report. That report does far more than state whether a patient has cancer. It identifies the exact tumor subtype, reveals the molecular drivers responsible for its growth, predicts how aggressively it is likely to behave, estimates prognosis, identifies targeted therapies, and helps physicians design an individualized treatment strategy tailored specifically to that patient's disease. This is the true meaning of precision medicine. Rather than treating every patient with the same diagnosis in the same way, modern oncology treats the biology of each individual's tumor. Patients receive therapies matched to the unique molecular characteristics of their cancer, improving effectiveness while avoiding unnecessary toxicity. Modern cancer pathology is therefore no longer simply the science of diagnosing disease. It has become the central decision-making platform that connects microscopy, immunohistochemistry, molecular biology, genomics, artificial intelligence, and clinical medicine into one unified goal: delivering the right diagnosis, selecting the right treatment for the right patient at the right time. That integration is transforming cancer care and bringing us closer than ever to truly personalized oncology.